want to start off with a couple of slides just to try to explain why, my interest in coastal wetlands and seagrasses in the middle of a sort of a highly coral-driven work or hard-bottom kind of work. And one of the reasons it begins kind of evolutionarily, that there's this sort of shared evolution and evolutionary history of angiosperm seagrasses and mangroves with other lineages like coral reefs, fishes, not the corals themselves, of course, but the fishes, a lot of the decapods, foraminifera, mollusks, and even manatees have this sort of evolutionary history. In addition to that, the other reason that these systems are linked is that tropically they're linked between seagrass beds, coral reefs, and coastal wetlands, such as you see here in Kaneohe Bay. And that would also be tropical wetland systems in other islands that would be dominated by mangroves. And finally, there's also a shared geological succession in these different systems. And I use Kaneohe and Kailua bays as an example of two sister bays, right, separated by Mokapu. But here you have the older, more senescent bay of Kailua, and that's the reef right now. And here's the old bay right in here, which is now a coastal wetland, Kawenue, and then Kaneohe Bay, which is a little bit more coral-dominated system than Kailua Bay right now. So these are some of the reasons I'm interested in these systems. Been very fortunate to be funded for 3 years in a row by HCRI, and I really appreciate that funding and support. So I thought I'd kind of come along and talk about the projects that you've funded without me really sort of the mortar between the bricks of the three proposals I had. These are the sort of areas that I was interested in and you kind of funded because the resources were there and it kind of all fit together, and I had 3 years in a row and I could sort of put these things together in those 3-year periods. So a little bit about that. And one of the species I was really interested in is one that would be found along coastal wetlands, and that's the seagrass Rupia maritima. It hasn't gotten a whole lot of attention here in Hawaii, and one of the reasons is it turns out it's quite a rare plant, and I'll talk about that in a minute. But it's a submerged plant. It's a flowering plant. Here it is with the endemic shrimp, Opaiula. It's a cosmopolitan species. It's found throughout the world. It's found in both temperate and tropical latitudes, both hemispheres, and it's extremely euryhaline, from 0 to 140 parts per thousand it can live at there. One of the key questions that people have had is it really one species across this very wide range, or are we talking about multiple species, just poorly understood species complex? And I like the isolation of Hawaii. It's a good place to start. It's found here. So we look at that. And the other reason I want to look at it is we are committed to manage to preserve endemism. So if it turned out that our Hawaiian Rupia was a little bit different than, let's say, the mainland of North America, we'd want to know that and apply that into our management concepts here. And so I've got biogeographic collections from North America as well as from Hawaii. And I did want to note I've been to all 5 of the main Hawaiian islands, and I've been out to about 52 sites, but only found Rupia at 15 sites, which is pitifully low. And again, I'll talk about that in a minute. I'm only going to talk about the chloroplast sequences that I have here, the results of that, just very, very briefly. I know we're pressed for time, but I used the chloroplast, so it wouldn't be the genes, but the spacers between the genes here. And I've looked at 5 different chloroplast regions. And this is the tree of the collections that I have. This is a concatenated tree, so this is all 5 markers put together. And it's Bayesian and about 3,800 base pairs. And you see right away, if we can look at the mainland here, the Atlantic and the Pacific clades, or well, not really clade, but the Pacific collections and the Atlantic collection. In Hawaii, we have 2 independent introductions, at least to the Hawaiian Islands, I believe, based on this work. And the ITS, which is another genome, I don't put the data in here today, but that supports this as well. So right away, what we see from the Rupia is that we likely have something different than the mainland, and that we even have maybe a little bit more diversity than the mainland here. So this is kind of interesting. Well, like I said, it turns out to be a rare plant. It shouldn't be that rare. There's a lot of coastal wetlands that have been set aside for waterbird management that DOFA manages. So you'd think that you could find it in there, and it actually should be there, but it isn't. So I'll continue with this question, but hopefully past my PhD, which hopefully will be soon. And I'll look at things like morphology, and I'll continue to look at sequence variation. I've gotten some specimens in, 2 different species in from New Zealand, and I'll get some other locations as well. But I really want to stress that it would be important to probably conserve this rupia here in Hawaii, since we seem to be different. Trying to understand a little bit about why this plant is so rare really leads you into the coastal wetlands, which I was funded for this year to go in and look at some questions. But I want to talk a little bit about invasive fish in Hawaii's coastal wetlands. It's somehow, I think it's kind of fallen through the cracks in some of the management issues, because the coastal wetlands are sort of managed by DOFA, Fish and Wildlife Service, and some other folks that are interested in the water birds, and they sort of focus on managing those systems for water birds. And then DAR, which is charged with managing more of a fish, they don't really get into these sites as much, and these sites are just chock full of invasive species. And I was particularly interested in seeing if potentially rupia is being lost in these systems because of these invasive fishes. And in fact, I think this is one of the primary culprits right here. This is the black-chin tilapia, Ceratherodon. And understanding that with these tilapia, all these tilapiid fishes, their evolutionary history is a little bit mixed. Understanding species boundaries is a little bit challenging. And so I'm working with Bishop Museum to try to understand that, and they're on board with this. But this is the tentative identification of this right now, this fish. And what we've done in a couple of places now, but I'll just go through one of the experiments. We have done things like transplant experiments of rupia into areas that the fish are present, but we sort of fence the area off and keep the fish out, and the rupia grows just fine. So it's not as if the rupia is not found in some of these sites because the water quality isn't adequate, or the sediment that they're anchored in wouldn't be adequate. I won't give those data today, but we do have those. Instead, we really want to, this is kind of like our smoking gun experiment, where these are pasta strainers, and we just invert them and make a cage and put a fragment of rupia in there along with an individual fish. And we looked at, with the tilapia, we looked at 2 different size classes, and then we had a procedural control, which are top minnows, which are another invasive fish here, and 2 different size classes, and then, without the fish as a control. And these are just the field photographs. I have them scanned in, but I had this slide ready, so I have to show you a little bit of the control, what the rupia fragment looks like, and before and after, it's no difference. And then with the small tilapia, before and after, no difference. And the same thing true with the top minnow that we used as the procedural control. But if you look at the large tilapias in here, that's before and that's after. The fish have just munched it right down and consumed it. And